Hello all my dear students, good afternoon to all of you. Welcome back once again to your online portal, that is what Biology at your fingers, on the behalf of Subrat Singh Children Academy. And as you know, I am like always Ajay Tiwari, your biology teacher, and welcoming you to all once again after a long period of time, because you all are aware about that. At present your offline classes are also running. So this video is just for the revision purpose. Whatever you just learn during the offline classes, the same topic will be revised here in the online mode, and this video or this series of the video will be totally for your board examination. And what are the certain important topics, what are the certain important questions that ask in your board examination from various chapter of your syllabus. And this video is really going to help, and after watching all these videos there will not be any doubt, as well as you will able to do 100% questions of your board exams. So this is just for the board exams, all those students who are just preparing for the board examination. So watch the complete video, and after watching the complete video you will able to clear all your concept related to particular chapter in just one video. Here today we are going to start the chapter number 2, that is what, sexual reproduction in flowering plants. You all aware about that chapter number 1 is deleted as per the CBSE new syllabus guidelines. So this is the chapter number 2, sexual reproduction in flowering plant, and this chapter will be completed in one video. And here we are going to discuss all the important topics, all the important questions those were asked in the previous examinations of the board, as well as in the various competitive examination too. So watch the complete video. So here let's see in this chapter what are the certain topics. The first, flower. We are going to discuss about the flowers and its various parts. Second, pre-fertilization structure and events. Okay. In pre-fertilization structure and events, we have to discuss about the what? First, development of male gametophyte. Development of male gametophyte. And this is very important for your board examination. यहाँ से क्वेश्चन बेटा पूछा जाता है. You are never going to skip this question. Development of male gametophyte. So in pre-fertilization structure event, development of male gametophyte is very important. Why this is so important? Because as you know, here we are going to form pollen grains. और pollen grains कैसे बनते हैं? What are the certain viability period of the pollen grains? What is pollen bank? All these questions will be discussed here. The second one is development of female gametophyte. So here in pre-fertilization structure and events, we are going to form the male as well as female gamete. The male gamete is what? Pollen grain, that is also known as the first cell of the male gametophyte. और दूसरा जो हमारा है बेटा female gamete, that is known as egg. And as you know, egg is present inside the ovule, and ovule is present inside the ovary. We will discuss later on. Next, the process of double fertilization. The process of double fertilization. The trend of this asking the question in the board examination is just from the pre-fertilization structure and event. Most of the questions are asked from here and were from asked here. And next, the double fertilization. Double fertilization is also much significant, because here, as you know, the fusion of the first male gamete with the egg resulting in the formation of zygote, and the second male gamete that will fuse with what? Two polar nuclei forming the triple R endosperm. So here in double fertilization, in the first step there will be formation of zygote with the fusion of the first male gamete, and the second male gamete will fuse with two polar nuclei forming the endosperm. So here fertilization takes place twice. That's why this is known as double fertilization. After this, we are going to discuss about the post-fertilization. Post-fertilization structure and events. So here we are discussing with pre-fertilization structure and events. So this is what? Very, very important. And from here, which topic is important? Development of male gametophyte and development of female gametophyte. And here, double fertilization itself is so important. Why? Because here always a particular question is asked. And again, the post-fertilization structure and events. Here we are going to discuss about the how endosperm formation takes place. How endosperm formation takes place. Here, what is endosperm? Then after, we will also discuss about the embryo formation in the case of both monocot as well as in the case of dicots. Here we will also discuss about the seed, and we will also discuss about the fruit. So if we consider all these events, so here the development of the embryo is so important, because here there are certain structures, those are also given in your NCERT, and from there how the development of the embryo takes place in the monocot and how development of the embryo takes place in the dicot. All these topics are so important. And remember here seed is also important, because as you know, this is the end product of the sexual reproduction. Okay, and seeds, category of the seeds are also so important. Fruits, as you know, ovary develops into fruit, and here ovule develop into seed. We will discuss all those fate of the various structures in the post-fertilization events, means that which part is developing from which part of the female gametophyte. अगर हम देखेंगे कि फीमेल का जो gametophyte है उसके कौन से पार्ट से जो हमारा सीड या फ्रूट बन रहा है या सीड कोट बन रहा है, किस पार्ट से बन रहा है, उनकी प्लॉइडी लेवल क्या है. तो all these questions are so important. So these are the some technical things. Here in the last we are going to discuss about the apomixis. As you aware about that, Apomixis is a type of asexual reproduction that mimics the sexual reproduction. So how Apomixis seeds are formed, this is very, very important. So from here, this Apomixis is very, very important. And then after here, we will also discuss about the embryony. So these are the two special events. Those should be discussed during the course of this chapter number 2. So I am going to erase all these topics, and remember, from all these topics, the maximum probability of the asking the questions, just seeing the trend of the board, is this from pre-fertilization structure and events, and then after post-fertilization structure and events. So these two topics are so important, or very important, or much more important for your board examination. Then after, another one is that is what double fertilization is also as it is important, but maximum long answer type question will be asked from the pre and post fertilization structure and event. So without wasting the time, we will start with pre fertilization structure and event, and in that we are going to form the male as well as female gametophyte along with the structure of the flower. If we are going to discuss the structure of the flower, you all studied in your class 11th. Okay, so structure of the flower we will quickly recap, and then after here in the flower, as you know, there are some accessory structure or accessory holes, and some there are essential holes. In essential holes, there is a male and female reproductive organs, respectively stamen and pistil, those are situated, and in the, we can say that accessory holes, there is a sepal and petal. So all these points are not as much important, but we will start with the flower, and then after we will discuss about the pre-fertilization structure and events and all these topics one by one. Okay, dear students, so we are starting with flower. As you know, flower is the site of sexual reproduction. Okay, this is site of sexual reproduction, and flower is a what? Modified suit. Here we will discuss only important things. So these 2 points are important, that flower is the site of sexual reproduction. It means that sexual reproduction will take place here, and flower is known as modified suit. You all aware about this because you have studied in the morphology of the flowering plants in class 11th. Let's take a quick recap. Why this is known as modified suit? Let's discuss that. Let's consider this is what? Apical meristem, and these are the internodes. From here there is arrangement of female reproductive part. Here there is attachment of what? Stamens. These are the petals, and these are the sepals. When we will compress all these structure, we will see such kind of structure, and this one is known as what? Thalamus, or we can also say this is what? Receptacle. Okay, so we will not discuss more about these points because these are not much significant, and here we are discussing with only important things. This is just for a revision. So flower is what? Site of sexual reproduction, and this is what? Modified suit. Okay, let's discuss from here. Flower contains 2 essential holes, and these 2 essential holes we will see stamen, and we are also going to observe the pistil. Okay, stamen is male reproductive part, and pistil is what? Female reproductive part. Okay, so let's consider here stamen, that is also known as what? Male reproductive part. It has 2 parts, those are anther, and the second one is filament. While if we consider the second one is pistil contains stigma, that is second one is style, and the third one is ovary. What are the function of this? Stamen, this contains anther, and in anther we will discuss that how pollen grains are formed, and here stigma, this is the site of landing of the pollen grains. Here after the process of pollination, pollen grains landed here. Style, this is just simply helping structure, and it attaches the stigma to the ovary, and ovary, as you know, that this is the solen part of the flower, and here there is a presence of ovule, and inside the ovule there is a presence of egg. So this was all about. Let's discuss all these things. And second, these are the 2 essential holes. Now we will discuss accessory holes. In accessory holes we will discuss about the sepal. Sepal is usually green in color. It is what? Green color, and in general we can write responsible for protection. Okay, beta. And the last one is petal. Petal is what? This is bright color portion. And why this is bright color? To attract the insect for the pollination purpose. So all these were the essential and non-essential holes of a flower. And remember, if all the sepals we constitute, so all the sepals together forms, as you know, calyx, and all the petals together form कोरोला. कहने का मतलब जब हम सारे के सारे सिपल्स जितने हैं उनको इकट्ठा करके कलेक्टिवली बोलेंगे तो उनको हम बोलेंगे कैलिक्स, और जितने हमारे पेटल्स हैं उनको एक साथ हम क्या बोलते हैं? कोरोला बोलते हैं. तो ये सारी चीज़ें थी, इसको अब एक डायग्राम के माध्यम से आपको बताते हैं. लेट्स कंसीडर अ पर्टिकुलर डायग्राम, एंड बाय मींस ऑफ दैट यू विल एबल टू क्लियर इट. सो फर्स्ट ऑफ ऑल, हियर वी आर जस्ट मेकिंग व्हाट? दिस इज व्हाट यू ऑल अवेयर अबाउट दैट? Thalamus. And here, as you know, there is a solen part, and that solen part is known as ovary, and here there is female reproductive part of the flower. This one is known as what? Stigma. This one is known as what? Style. This one is known as what? ovary, and inside the ovary there is a presence of ovule. Okay, I am going to erase this, and after erasing this you will able to just observe rest of the parts of the flower too. Okay beta, so I am doing it fast. Here this is the filament and this is what? Anther. Let's make one more. Numbers are not fixed, you can make as per your wish. This is what? Anther and filament, and again here there is what? Anther and filament. Okay, so we will write it what? Anther, we will write it as filament, and now here there will be what? We will observe here petals. These are the what? Petals. We will make it here also, and there are certain what? Sepals. So these are what? Sepals. Those are collectively known as calyx, and these are what? Petals, collectively known as corolla. This is what? थैलमस, और नीचे की तरफ जो है बेटा ये पेडिसिल है. ठीक है. सो दिस इज व्हाट? अ टिपिकल स्ट्रक्चर ऑफ द फ्लावर. इन टिपिकल स्ट्रक्चर ऑफ द फ्लावर वी आर गोइंग टू फाइंड आउट द sepals, petals as a non-essential holes or accessory holes, and here anther, and along with anther and filament together these two forms stamen. Anther and filament together form stamen, and stamen, as you know, male reproductive part, and here stigma, style and ovary together forms pistil, and pistil is known as female reproductive part of flower. So this was all about flower. Remember, outside this there will not be any question, those may be asked in your board examination, so you all aware about that. So now we are going to discuss about the detailed structure of the male reproductive part of the flower and female reproductive part of the flower, and from here a particular question may be asked. So I am going to erase this, and after that we will start. Okay dear students, so let's start with male reproductive part of flower and its structure. Okay, from here this is very triple star question, most important. Definitely a particular question may be asked from here. Let's discuss. As we know, these are the what? Here these are the filament. Okay, we earlier discussed these are the what? Anther, and these two collectively known as stamen, and stamen is known as male reproductive part. And here there is a sterile tissue that is known as connective, that is present, and you can see from here anther is bilobe. Let's discuss a detailed structure here. So if we cut the section, we will see such kind of a structure, and as you know, here anther is what? Bilobed, so we can make it like this, and this will be okay. So here these two are the what? Lobes. Okay, this is what? यहाँ पे हम कनेक्टिव नहीं बनाते बेटा, कनेक्टिव यहाँ पे होगा. This is what? Connective. So from this, what is significant thing? And these are what? Microsporangia, in which microsporangia or sporangium in which microspores will be formed. Okay, so from here how the question will be asked? Question is here, the anther is what? Dithecous. What? Dithecous. It means that how many theca? 2 theca, and theca means that 2 lobes, and here there are 2 theca as well as 2 lobes. How? Let's find out. Here this is the lobe number 1 and this is the lobe number 2, and inside each lobe there is what? 2 pollen sacs. Okay, inside these 2 lobes there are 2 pollen sacs. So total how many pollen sacs? 4 pollen sacs in particular anther. So total how many pollen sacs? 4 pollen sacs. But this structure is not as many as significant. Okay, so अब कहाँ से पूछा जाएगा बेटा? क्वेश्चन पूछा जाता है TS of anther. Okay, so this question is like that. Your oxygen, without oxygen you cannot survive, and this is what? Something like that. It is much significant. Okay, so let's discuss about the transverse section of the anther. Okay, so let's see TS of anther. This is much important question, इसको आपको भूलना नहीं है बेटा. As we discuss, कैसे बनाएंगे हम? Like this. Okay, and like this. Okay, how many layers? There are 4 layers. All those layers, the first one is epidermis. Epidermis is what? Single layered. What is the function of this? Protection. What is the function of this? Protection. After that, endothecium. This is also what? Single layered and have callose thickening. This is important, and have callose thickening, and this callose thickening will hold the microspore tetrad. That is much important. The third is what? middle layer. Middle layer, it is made up of 2 to 4 layer of parenchymatous cells, and this is also protective. So we can say that collectively that epidermis, endothecium, this one is epidermis, this one is endothecium, and this one is what? Middle layer. All these are protective. यानी तीनों के तीनों क्या हैं? Protective है. And the last layer, that is known as what? Tapetum. This is what? Nutritive. Means that provide nourishment. It will provide nourishment to developing माइक्रोस्पोर, यानी जो माइक्रोस्पोर मदर सेल के अंदर डेवलप कर रहे हैं उनको नरिशमेंट प्रोवाइड करने का काम टैपेटम करती है. क्वेश्चन ये कई बार पूछा गया, व्हाट इज टैपेटम? टैपेटम इज द इनरमोस्ट लेयर ऑफ anther. What is the function of this? It is responsible for nourishment as well as it also produce UVS bodies. UVS bodies को produce करती है, and those UVS bodies just going to help in the synthesis of sporopollenin, and that will form the exine. Next, this is also responsible for releasing the pollen kit. Pollen kit means that this is the protective structure, protection from the UV rays, as well as we can say that it also help the adherence of the pollen grains to the insect or animals. कहने का मतलब ये है टैपेटम का एक फंक्शन न्यूट्रिशन का है. इसके साथ साथ यूविस बॉडी फिर क्या करती है? स्पोरोपोलेनिन की सिंथेसिस करती है, जो कि एक्साइन बनाएगा आगे चल के पॉलन ग्रेन का. इसके साथ साथ बेटा ये पॉलन किट पूछा गया, व्हाट इज पॉलन किट? तो पॉलन किट इज ए व्हाट? येलो पाउडरी मटेरियल. आपने देखा होगा कुछ ऐसे फ्लावर जैसे चाइना रोज या हिबिस्कस हैं जो आपके हाथों पे लगे तो कुछ ऐसा मटेरियल होता है जो आपके हाथों पे लग जाता है. तो एक्चुअल में वही पॉलन किट होता है. पॉलन किट का फंक्शन क्या है बेटा? ये जो आपके इंसेक्ट हैं आते हैं फ्लावर पर बैठते हैं, किसके लिए? पॉलन ग्रेन्स के लिए या नेक्टर के लिए, तो उसकी बॉडी पे चिपक जाते हैं और फिर पॉलन ग्रेन्स इजीली कैरीड हो जाते हैं. इसके साथ साथ दे आल्सो प्रोटेक्ट द प्लांट फ्रॉम द यूवी रेडिएशन. सो दे आर प्रोटेक्टिव एज वेल एज हेल्पिंग इन द पॉलिनेशन प्रोसेस. सो ऑल दोज पॉइंट्स वी आर नॉट राइटिंग हियर. Tapetum, nutritive, as well as produce UVS bodies, as well as also secreting the pollen kit. So let's make all those four layers. Here, as you know, there is a sterile tissue that is known as what? Connective, and here there will be presence of microsporangium. So all these are what? Microsporangium, and this is microsporangia in the case of young anther, and in the case of mature anther it will converted into pollen sac. ठीक है बेटा. तो हम देख सकते हैं these are the two lobes. ठीक है, दो लोब दिख रहे हैं और दोनों में हमको क्या क्या दिख रहे हैं? दो pollen sac दिख रहे हैं. इसके अंदर बेटा सेल्स भरी पड़ी होती हैं, and those cells are compactly arranged, and these are known as what? Archisporial सेल्स. इनको हम क्या बोलते हैं? आर्कीस्पोरियल सेल्स. और अगर हम यहाँ से यहाँ तक की डिस्कस करें डिस्टेंस की, तो इसे हम बोलते हैं एंथर wall. So anther wall has how many layers? 4 layers. Those are epidermis, endothecium, middle layer, and this one is tapetum. So let's quickly discuss all those 4 layers. Here we will make the first, that is outermost, that is what? Epidermis. This is what? Simply flat cells. I am not making here full. I will make the half portion, और आप आधा खुद से बनाएंगे. ठीक है बेटा. तो हमने आधे में बना दिया. इसको हम क्या कहेंगे? This is what? This is what? Epidermis. Epidermis के बाद बेटा क्या होगा? अगली सेल्स आपकी रहेंगी कौन सी? एपिडर्मिस के बाद ये जो सेल आपको दिख रही है बेटा, दूसरी लेयर हम बना रहे हैं. दिस वन इज व्हाट? इंडोथीसियम. और इंडोथीसियम के पास क्या होती है? कैलोस थिकनिंग. तो दिस इज व्हाट? इंडोथीसियम. और इंडोथीसियम के पास क्या है बीच बीच में? कैलोस थिकनिंग. उसके बाद मैंने बताया था कि मिडिल लेयर होती है. मिडिल लेयर 3 से 4 लेयर की हो सकती है, मतलब यहाँ पे सेल्स 2 या 3 या 4 लेयर्स में हो सकती हैं. तो वो मैं यहाँ पे आपको बना के दिखा रहा हूँ. ठीक है बेटा, ये फटाफट आप यहाँ से बना सकते हैं 2 से 4 लेयर्स. और ये बन के तैयार हुआ. इसको हमने क्या कहा? मिडिल लेयर. दिस आर व्हाट? मिडिल लेयर. एंड द लास्ट द टैपेटम. टैपेटम की जो सेल्स होती है बेटा वो कोनिकल होती हैं कुछ इस तरह से. किस तरह से? कोनिकल, लाइक दीज. तो हम यहाँ से कोनिकल सेल्स बना रहे हैं बेटा, तो फटाफट बनाते हैं. दीज आर द कोनिकल सेल्स. और ये फटाफट यहाँ से बन रहा है. दीज आर द व्हाट? द इनर मोस्ट, दैट इज व्हाट? टैपेटम. टैपेटम में भी मल्टी न्यूक्लिएट. दिस क्वेश्चन में भी आस्क हाउ टैपेटम बिकम मल्टी न्यूक्लिएट. तो टैपेटम बिकम मल्टी न्यूक्लिएट बाय द amitosis, yeah, we can write here endomitosis, and the second one is what? Free nuclear division. इसका मतलब क्या हुआ? अगर एंडोमाइटोसिस हो गई या केवल न्यूक्लियर डिवीजन हुआ और साइटोप्लाज्म का डिवीजन नहीं हुआ, तो इस कंडीशन में भी टैपेटम जो है उसकी जो प्लॉइडी है वो मल्टी न्यूक्लिएट होने वाली है. ठीक है बेटा. अब इसी के अंदर मैंने यहाँ पे जो सेल्स बनी थी, मास ऑफ द सेल्स हैं जिनको हम बोलते हैं आर्कीस्पोरियल सेल्स. सो दिस इज व्हाट हमने बनाया यहाँ पे यंग एंथर. रिमेंबर, इफ यू आर गोइंग टू ड्रॉ द डायग्राम इन द बोर्ड, मैंने एक तरफ बनाया है, दूसरी तरफ ऐसा ही रहेगा. यू ऑल हैव टू मेक एट योर ओन. ओके, सो दिस इज व्हाट बेटा? दिस इज कनेक्टिव. ओके, और आज की डेट कौन सी है आप सभी को मालूम है. टुडे इज व्हाट? 5 मार्च 2021. सो ये वीडियो आपकी रिवीजन पर्पस से है. तो अभी तक हमने क्या देख लिया बेटा? अभी तक हमने देखा एंथर, और उसी एंथर में क्या बनने वाला है? मेल गैमीट बनने वाला है. तो हमारा अगला टॉपिक है व्हाट? Development of male gametophyte. So before that, let's quickly recap the function of the tapetum. Nutritive है, also producing what? यूबिस बॉडीज, एंड यूबिस बॉडीज विल लेटर ऑन फॉर्म स्पोरो पॉलिन की सिंथेसिस में मदद करेंगी, और वो क्या बना लेगी, एग्जाइन बना लेगी किसकी, पॉलन ग्रेन की। इट ऑल्सो सिक्रेट व्हाट, पॉलन किट भी ये सिक्रिएट करती है बेटा। एक पर्टिकुलर एंजाइम भी रिलीज करती है जिसका नाम होता है कैलेज। तो कैलेज एंजाइम मैंने बताया था, यहाँ पे देखिए, यहाँ पे आप देख सकते हैं इंडोथीसियम, यहाँ पे जो कैलोज की थिकनिंग थी जिसके द्वारा माइक्रोस्पोर टेट्राएड जो आपस में जुड़े थे उनको ये रिलीज कर देगा कैलेज एंजाइम। तो ये 4-5 फंक्शन है टैपेटम के, आपको याद करने हैं, बहुत इंपॉर्टेंट क्वेश्चन है बेटा। और ये ट्रांसवर्स सेक्शन ऑफ एंथर, ये भी बहुत इंपॉर्टेंट है, जो भी समय आपके बोर्ड पे दिख रहा है, दिस इज मच इंपॉर्टेंट, एंड रिमेंबर फ्रॉम हियर अ पर्टिकुलर क्वेश्चन विल बी आस्क्ड इन योर बोर्ड एग्जामिनेशन। ठीक है, सो नाउ वी आर गोइंग टू मूव स्ट्रेट फॉरवर्ड टुवर्ड्स द डेवलपमेंट ऑफ मेल गैमेटो। ओके बेटा, तो हमारा अगला टॉपिक है डेवलपमेंट ऑफ मेल gametophyte. Development of male gametophyte. This is what most important questions. How? Up till now we have discussed that this was microsporangium, in which these were the archisporial cells, and these microsporangium was present inside the anther. We have discussed in the transverse section. So this is what microsporangium. Okay, the microsporangia कहाँ है? In the case of young anther. Okay, and inside these are what, archisporial cells. Okay, when anther mature, here the cells arrangement will be like this, they will loosely arranged. Okay, so now these are known as pollen sacs, and pollen sacs in the case of mature anther. Okay, here we will see in the case of mature anther में हमने देख लिया। And now from here any one of the cell will act as a what, microspore mother cell, and these microspore mother cell will be diploid. Okay, and rest of these cells will be degenerated. Okay, and here there will be what, a meiotic division. कौन सा division होगा, meiotic division, and meiotic division will result in the formation of microspore tetrad. This is what, microspore tetrad. Okay, and remember these microspore tetrad are attached with each other with the help of callose thickening. Those were formed by the second layer endothecium. Okay, so this is what, microspore tetrad, because these are the result of the meiotic division. We will discuss the ploidy level again. Here microspore tetrad may be in a certain arrangement. If these are like this, tetrahedral, it may be like these. So if the arrangement is like this, they may be termed as what, linear, and there are certain more possibilities, but those are not part of our board examination. So here up till now we have formed microspore tetrad, and from this microspore tetrad callose thickening will be dissolved by the what, callase enzyme, and after this there will be formation of 4 microspore. All these are what, microspore, and all these microspores are haploid. Okay, and now next here कौन, टैपेटम बेटा रिलीज करेगी स्पोरोपोलिन को, और किसी एक माइक्रोस्पोर को हम यहाँ बनाते हैं। सो दिस वाज द माइक्रोस्पोर, इनसाइड दिस देयर वाज अ न्यूक्लियस, दिस इज व्हाट, प्लाज्मा मेम्ब्रेन। इसके बाहर एक और लेयर होगी बेटा जिसको आप क्या बोलते हो, इंटाइन, और आपको मालूम है इंटाइन किससे बनती है बेटा, सेलुलोज, और उसके साथ साथ कौन होता है, पेक्टिन। ठीक है, बाहर एक और कवरिंग होगी जिसको आप क्या बोलोगे, जिसको हम बोलेंगे एक्साइन। क्या बोलेंगे, एक्साइन बोलेंगे। ठीक है, तो ये मैंने जो बनाया, दिस इज व्हाट, exine, and exine is made up of sporopollenin, that is most resistant material. Okay, or most resistant organic material. These are the what, germ pore. And now this structure is known as what, pollen grain. This structure is known as what, pollen grain, and pollen grain is the first cell of male gametophyte. So what is the difference between microspore and pollen grain? Microspores are the structure, those are the result of the meiotic division without the exine, and if intine and exines are secreted, now this microspore will be termed as what, pollen grain. And in the case of most of the angiosperm, this pollen grain will undergo mitotic division. कौन सा division जाएगा बेटा, mitotic division, resulting in the formation of 2 cells like this, one larger one and one smaller one, and outside this, like always, there will be exine and there will be what, germ pores too. This larger one is known as what, vegetative, इसको क्या बोलेंगे, वेजिटेटिव सेल, और दिस इज ऑल्सो नोन एज ट्यूब सेल। एंड और ये ट्यूब सेल क्या करेगी बेटा, आगे चल के पॉलन ट्यूब बनाएगी। ठीक है, ये आपकी कौन सी है, जनरेटिव सेल, और जनरेटिव सेल क्या करेगी बेटा, मेल गैमीट बनाएगी। क्या बनाएगी, मेल गैमीट। कितने, 2, किस डिवीजन से, माइटोटिक डिवीजन से। ओके बेटा, तो ये 60% केसेस में बेटा ये 2 सेल स्टेज में कौन आपका पॉलन ग्रेन सेट ऑफ हो जाता है। तो क्वेश्चन पूछा गया इन द मेजॉरिटी ऑफ द एंजियोस्पर्म 60% ऑफ द एंजियोस्पर्म पॉलन ग्रेन्स आर सेट ऑफ एट द 2 सेल स्टेज। तो दोज 2 सेल्स आर वेजिटेटिव एंड जनरेटिव, बट इन द रेस्ट 40% हियर दिस जनरेटिव सेल विल डिवाइड एंड इट विल फॉर्म 2 मेल गैमीट, सो 2 मेल गैमीट प्लस 1 ट्यूब न्यूक्लियस, सो देयर विल बी व्हाट टोटल 3 सेल्स। ओके, तो उसको हम बोलते हैं 3 सेल स्टेज। वो कब होगा? जब पॉलन ग्रेन जाके स्टिग्मा पे लैंड करेगा, पॉलन ट्यूब का फॉर्मेशन होगा, फिर उस पॉलन ट्यूब में माइटोसिस होगी और उसके बाद 2 मेल गैमीट बनेंगे। तो अगला स्टेज हम देखेंगे यहाँ से, ये 2 सेल स्टेज वाला है, ये हमारा कौन कौन सी वेजिटेटिव और जनरेटिव सेल है। अब जैसा मैंने बताया, ये पॉलन ग्रेन जाएगा, लैंड करेगा स्टिग्मा पे, लैंड करने के बाद ये जनरेटिव सेल डिवाइड होगी कब? जब पॉलन ट्यूब बन जाएगी। तो हम यहाँ से देखते हैं, मान के चलते हैं ये हमारी कौन सी होती है? वेजिटेटिव सेल। इसने बनाने का काम किया हुआ है पॉलन ट्यूब, तो ये हमारा पॉलन ट्यूब बन रही है, ठीक है बेटा। और इसके बाहर लाइक ऑलवेज एक्साइन होने वाली है। यहाँ पे क्या होंगे? वैक्यूल्स होंगे, दीज आर वॉल वैक्यूल्स। ये सेल डिवाइड हो चुकी है और इसने क्या बना लिया है? 2 मेल गैमीट के साथ साथ 1 ट्यूब न्यूक्लियस। तो ये दोनों क्या हैं? 2 मेल गैमीट और उसके साथ साथ ये क्या है आपका? ट्यूब न्यूक्लियस, ठीक है। इसको थोड़ा सा अगर आप बड़ा करना चाहें तो कुछ इस तरह से आप बना सकते हैं, ठीक है। ये आपका 2 मेल गैमीट, ये ट्यूब न्यूक्लियस, यहाँ पे कौन? वैक्यूल्स और उसके बाहर कौन? एक्साइन, और इसी को क्या बोलते हो आप? मेल गैमेटोफाइट। इसको हमने क्या बोला? मेल गैमेटोफाइट। सो मेल गैमेटोफाइट का मतलब, और गैमेटोफाइट एक्चुअल में होता क्या है? गैमेटोफाइट मींस दैट द स्ट्रक्चर दैट हैज द गैमीट्स। तो इसके पास देख सकते हैं ना 2 मेल गैमीट है, इस वजह से ये क्या है? मेल गैमेटोफाइट है। तो दिस वाज ऑल अबाउट द डेवलपमेंट ऑफ मेल गैमेटोफाइट। एक बार फिर से देख लीजिए बेटा, यहाँ से ये माइक्रोस्पोरेंजियम है जो कि प्रेजेंट थी किसके? एंथर के अंदर। कहाँ? एक लोब में 2, इसमें आर्किस्पोरियल सेल्स थी। ये आर्किस्पोरियल सेल्स आगे चल के एक्ट करती हैं एज ए माइक्रोस्पोर मदर सेल। उसके पहले ये यंग थी, तो माइक्रोस्पोरेंजियम मैच्योर हुई तो क्या हो गया? पॉलन सैक। इस पॉलन सैक की कोई भी एक सेल माइक्रोस्पोर मदर सेल की तरह काम करेगी। नाउ देयर विल बी व्हाट? meiotic cell division forming the microspore tetrad. All these microspores will be separated by the Gallage enzyme. Then after there will be secretion of the exine and intine. Now this microspore structure will change to pollen grain, and now pollen grain by means of the mitotic division will form 2 cells, vegetative cell and generative cell. Vegetative cell will form pollen tube and generative cell will further divide to form the 2 male gametes, resulting in this structure here. These are the what? Vacuoles. These are the vacuoles and this is what? Pollen tube that contains 2 male gamete and 1 tube nucleus. ये large portion है बेटा, यहाँ पे vacuoles, ये exine है, ये 2 male gamete और एक ये tube nucleus है। तो this was all about the development of the male gametophyte। अब अगले क्वेश्चन जो यहाँ से बनते हैं, ये तो ये क्वेश्चन यहाँ से बनता है डेफिनेटली। नेक्स्ट क्वेश्चन कैसे बनता है वो आपको ओके बेटा। अब यहाँ से देखते हैं pollen grains के बारे में थोड़ा सा। यहाँ से ये क्वेश्चन इंपॉर्टेंट है आपके लिए बेटा, pollen grains। और pollen grains में हम देखेंगे ये होते क्या हैं। These are the first cell of male gametophyte. Pollen grains are the first cell of male gametophyte. It means that male gametophyte will be developed from these later on. They contain exine and intine, and along with there is a what? Nucleus. Okay. Certain pollen grains are edible. Pollen grains are edible. Edible होते हैं बेटा. Hence used in syrups, tablets, for stamina boosting for children and athlete, as well as we can also write race horses, ठीक है। रेस वाले घोड़े के लिए, एथलीट्स के लिए, बच्चों के लिए ये स्टेमिना बूस्टर का काम करते हैं, ठीक है। ये इंपॉर्टेंट है। देयर आर सर्टेन pollen grains also lead to allergy and that condition is known as hay fever। यानी pollen grain के वजह से एलर्जी होती है जिसको hay fever कहते हैं। फॉर एग्जांपल आप देख सकते हैं पार्थिनियम जिसको आप दूब ग्रास भी कहते हैं, एंड दिस इज व्हाट? एलर्जिक। ओके। नेक्स्ट क्वेश्चन यहाँ से पूछा जाता है बेटा, pollen viability means that ability of the pollen grain to land on the stigma and then germinate। ओके। मतलब ऐसी यहाँ से एबिलिटी पॉलन ग्रेन की कि वो स्टिग्मा पे लैंड करके जर्मिनेट कर सकें। या हम लिख सकते हैं टाइम पीरियड ऑफ द पॉलन ग्रेन्स इन विच दे कैन लैंड ऑन द स्टिग्मा एंड जर्मिनेट। फॉर एग्जांपल वी कैन राइट द राइस, व्हीट, ये इनके बेटा 30 मिनट की वायबिलिटी होती है। वायबिलिटी यहाँ पे आपको कम देखने को मिलेगी। दूसरा अगर आप देखें लेग्यूम्स में आपको देख सकती है सेवरल मंथ में। ओके। एक जो लास्ट क्वेश्चन यहाँ से पूछा जाता है, पूछा भी गया है, व्हाट इज़ पॉलन बैंक। ओके। तो पॉलन बैंक क्या होते हैं? वी कैन cryopreserved pollen grain in liquid nitrogen at -196 डिग्री सेंटीग्रेड। ये क्वेश्चन आपके नीट में भी पूछा जा चुका है कि पॉलन ग्रेन्स को हम प्रिजर्व कर सकते हैं -196 डिग्री सेंटीग्रेड पे लिक्विड नाइट्रोजन में, एंड एक्ट एज पॉलन बैंक। और पॉलन बैंक का हम यूज़ करते हैं यूज्ड इन साइंटिफिक रिसर्च वगैरह में हम करते हैं। कुछ चीज़ें आपको और बताते हैं बेटा। जो आपका लार्जेस्ट पॉलन ग्रेन होता है, लार्जेस्ट पॉलन ग्रेन मिराबिलिस का है, मिराबिलिस। ओके, स्मॉलेस्ट जो है आपको दिखेगा मायो सॉटिस में, और नीडल लाइक दिखेगा आपको जोस्टेरा में। Okay, so this was all about the male reproductive part of the flower and their structures as well as development of male gametophyte. So the next topic is female reproductive part and development of female gametophyte. Okay students, so until now we have discussed about the male reproductive part and their structure as well as how the male gametes are produced in the topic development of male gametophyte. So it's time to study about the development of female gametophyte as well as how female gametes are formed, what are the certain structures those are involved in the formation of female gametophyte. So without wasting the time, there are two more important things related to the previous topic. Those are already written on your whiteboard. Generally we have seen that in a particular flower there are sepals, petals, gynosium, androsium, all these structures are present. But if the sepals and petals, these cannot be differentiated. Generally petals and sepals both are differentiated, but if we are not able to differentiate the sepals and petal, that condition is known as what? Perianth, or that whole just represents what? Perianth. So this line is important, that if sepals and petals are not differentiated, that whole is known as what? Perianth. Okay, and each unit of that is known as tipple. तो अभी तक हमने sepal देखा था, एक tipple भी होता है. What is tipple? If sepals and petals are not differentiated, that whole is called perianth, and each unit of that whole is known as what? Tipple. So now we are starting with female reproductive parts. So here we will write pistil, megasporangium, or we can write ovule. Okay, so this is the exact line of your NCERT. So first of all, here the female, we can write here the female reproductive, whole of plant is what? Representing what? Gynosium. Okay, so what is the term gynosium? Gynosium means that female reproductive whole of plant, and this gynosium is made up of one or more pistil, or we can write carpel. So this line is very, very important here. How? From here a particular questions may be asked, and in the previous examination questions were the part of your board examinations. So here we are going to write gynosium, and there are two possibilities. If there is only one pistil, that condition is known as, चूंकि क्या है, एक pistil है, that's why mono pistil is also known as carpel, hence monocarpillary. That condition is known as monocarpillary. If more than one pistil, that condition will be known as, more than one means that multi, and pistil, or we can write here carpel, that's why carpillary. Okay, so simple. Monocarpillary means that one pistil. If more than one pistil, that condition is known as multicarpillary, and if it is multicarpillary, then again it may be apocarpus. Apocarpus means that if there are more than one pistil or carpel, both are free. वो क्या है बेटा? Free है. Okay, second condition is here what? Syncarpus. It is clear from its name, there is something that is sync, मतलब fuse है यहाँ पे. So here more than one carpel or pistils are present, those are fused with each other. So here this is a fused condition, here there is a free condition. Now there is a trick to remember these two. There are two examples, those are given in NCERT. So how? Let's consider the first alphabet M, that for multicarpillary, second alphabet A for apocarpus, and the example of NCERT, that is Michaelia. तो आप कौन सा कोड याद रखेंगे? कोड रहेगा आपका मैम, और एग्जाम्पल हो जाएगा आपका Michaelia. Okay, same here. M stand for multicarpillary, S stand for syncarpus, and P that is given in your NCERT. The code is here what? MSP, means that here we are discussing with pi power. So this question is very, very important for your examination point of view. So here this was all about the gynosium. Okay, we can write here the female reproductive unit contains divided into three parts. There are three parts. what are those parts? Let's quickly make it. We have already mentioned during the structure of the flower. This is what? Stigma. This is what? Style. And this is what? Ovary. And inside the ovary there is what? Ovule. Okay, so stigma is the landing platform for the pollen grains. Style is a long cylindrical structure that attaches the stigma to the ovary, and ovary is the basal, solen or bulge part of the female reproductive system, or we can write female reproductive part of the flower. And this ovary has ovarian cavity. Ovary has what? Ovarian cavity. And ovary has ovarian cavity, and that cavity is known as what? लोक्यूल. यानी ओवरी के पास क्या होती है बेटा? ओवेरियन कैविटी. ओवेरियन कैविटी को हम क्या कहते हैं? लोक्यूल. और इसके पास क्या होता है? प्लेसेंटा. एंड फ्रॉम प्लेसेंटा व्हाट? ओव्यूल्स, और वी कैन राइट मेगास्पोरेंजियम arises. Okay, so let's quickly discuss once more. Ovaries has ovarian cavity. Ovarian cavity is known as locules. Locules has placenta, and from here ovule or megasporangium will arise. So these are the lines, exact lines of your NCERT. And remember, in your board examination, most of the questions, nearly all the questions will be asked from your NCERT. So we are going to clear all the topics of the NCERT. We are not discussing here the broad syllabus. We are just focusing towards the board examination. That's why here there is exact pinpoint revision, means that exact line to line revision of the NCERT. So you are able to understood. Now we are straightforward moving towards the next topic, that is what? Megasporangium. From here we will already draw the diagram of the megasporangium or ovule. That's why we will save the time, and after that we will elaborate and explain all the topics. So I am going to draw the diagram first, then there will be next part of the explanation. Okay बच्चों, तो यहाँ से हमने लिख लिया है already ovule. Ovule is what? This is also known as integumented megasporangium that will later on form megaspore. So this is important. Already I have written in a particular megasporangium, or we can say that ovule, there are as many as first, that is what? Funicle, hilum, integument, micropyle, nucellus and chalaza. As many as 6 parts I have mentioned here. Okay, so we will just find out all these parts along with the diagram. So before that there is a certain points, those should be remembered. That first one is that funicle. This is the point of attachment of ovule to the placenta. यानी placenta से एक ऐसा अटैचमेंट का साइट जो कि ओव्यूल को क्या करता है? अटैच कराता है. अगला हाइलम, पॉइंट वेयर बॉडी ऑफ द ओव्यूल अटैच टू फ्यूनिकल, यानी एक ऐसा पॉइंट जहां पे ओव्यूल की बॉडी, क्योंकि ओव्यूल में बेटा 2 रीजन्स होते हैं. ओव्यूल के पास 2 रीजन होता है. एक को हम कहते हैं क्या? स्टाक. क्या बोलते हैं बेटा? स्टाक. एंड अनदर वन इज नोन एज बॉडी रीजन. सो दिस इज द पॉइंट वेयर बॉडी ऑफ द ओव्यूल अटैच टू फ्यूनिकुलस. मतलब ये जो बॉडी वाला रीजन है किसका? ओव्यूल का. हाउ इट अटैचेस टू द here funiculus. Next, integument. Integument is the outermost protective part. That may be 2, that may be more than 2, and it may be absent too in the certain case of stem parasites. As well as the next one is micropyle. Micropyle is the opening through which pollen tube will enter during the process of fertilization. And the next one is that nucellus. Nucellus is the mass of the parenchymatous cells, and inside which there is a location of the embryo sac, and this nucellus is also nutritive. In certain cases this nucellus is completely consumed, but if this persists, that condition is known as perispheneric condition. We will discuss later on. And the last one is chalaza. What is chalaza? Chalaza is the basal part of the ovule, and from where integuments are arises. So we are going to discuss all these structure by means of a particular diagram. This is the ovarian wall. This is what? Ovarian wall, यानी wall of the ovary we can say that. This is what? इसको हम कह सकते हैं placenta. And इस placenta से कौन attach होगा? यहाँ से देखिए funiculus और funicle. So we are going to make a typical anatropous ovule here. So something like this, and from here again like this. Okay, so we are drawing here a particular typical anatropous ovule. There may be 2 integuments, and here is the same condition. There are 2 integuments, and inside this cavity is just filled with the mass of the parenchymatous cells. So we are going to make it like this. This is the mass of the parenchymatous cells, and inside which there is a location of the what? Embryo sac. In that there are 3 antipodals, 2 polar nuclei, 1 axil and 2 synergids. We will discuss later on. और इसको घेर के रखता है बेटा कौन? Parenchymatous cells. ठीक है. So we are just showing here LS of anatropous ovule. As you all aware about that, there are 6 type of ovules we have discussed in the previous classes. So here out of that we are just making one. This one is known as what? Placenta. This one is known as what? As you know, funicle. And here you can see this is the body region, this is the stalk portion. So this body is attached to this one by means of a structure that was known as hilum. We can see here, this is the hilum. These two are the what? Integuments. ये हमको जो दिख रहे हैं बेटा इंटैगमेंट्स दिख रहे हैं. This is what nucellus. This one is diploid. This one is also diploid. And here we are just getting embryo sacs. We will discuss about this. That is the female gametophyte later on, how this is formed, but not here. So this is what all about, and this portion is known as what? Chalaza. Chalaza means that this is the basal part, and from where there is a origin of the integuments, and this opening here is known as what? Micropyle. So now we can find out all the structures. This funicle, this one, प्लासेंटा, यहाँ से देख सकते हैं ओव्यूल को अटैच करा रहा है ओवेरियन वॉल से. हाइलम, द पॉइंट ऑफ अटैचमेंट ऑफ द स्टॉक एंड द बॉडी ऑफ द ओव्यूल. इंटैगमेंट्स, these two outer covering for protection. Micropyle, this opening. Chalaza, the next one above mentioned there, this one is chalaza from where integuments are arising, and this one is nucellus. Here we can observe nucellus, and these nucellus are just made up of the mass of the parenchymatous tissues or cells, and out of these any one cell will act as a what? Megaspore mother cell. So this diagram will be remain as it is, and we will discuss the next, that is what? Development of female gametophyte. So rest of the things will be erased, and we are going to discuss about the development of female gametophyte. So I am going to wipe up all these, then we will start development of female gametophyte. Okay बच्चों, तो यहाँ से हम स्टार्ट कर रहे हैं development of female gametophyte. Development of female gametophyte. This is very important for your board examination. From here definitely a particular question will be asked. So here as we discussed, this is the micropylar region, this is the mass of the nucellar cells. तो हम यहाँ से देख सकते हैं कोई भी एक सेल, इसको हमने क्या किया, ये एक्ट कर रहे हैं किस तरह से? मेगास्पोर मदर सेल की तरह से. So this the mass of the cells, out of these the cells those just towards the micropyle will act as what? Megaspore mother cell. The ploidy level of will be of which? 2. Okay, this will undergo meiotic division. This will undergo which type of division? Meiotic division, resulting in the formation of megaspore tetrad. 1, 2, 3 and 4. This is what? Megaspore tetrads. Out of these 3, those are towards the chalazal end, they will degenerate. So here 3 degenerate. Why these 3 are going to degenerate? Because during the degeneration process they will provide the nourishment to the developing megaspore. So here जो आपका ये functional है, ये जो degenerate हो रहे हैं इनसे इनको थोड़ा बहुत एनर्जी भी मिलेगी, और ये वाला आपका functional है. तो इसको हम यहाँ से देख सकते हैं. This is what? Functional megaspore. You have to remember the ploidy level, that is here n. Okay beta. Now this functional megaspore will undergo certain kind of division, a special kind of division, in which there will be karyokinesis first, and then after there will be cytokinesis. So here there will be 3 repeated nuclear division that will be termed as what? Karyokinesis. And after karyokinesis there will be cytokinesis. So first there is what? First माइटोटिक डिवीजन. फर्स्ट माइटोटिक डिवीजन वो कैसा है? न्यूक्लियर डिवीजन है बेटा. अभी तो यहाँ पे कितनी सेल्स थी? 1. ये इनलार्ज हो जाएगा बेटा. इनलार्ज होने के बाद ये आपका चलाजा. दिस वन इज माइक्रोपाइल. देयर विल बी फॉर्मेशन ऑफ हाउ मेनी सेल्स? 2 cells. ओके, तो दिस कंडीशन इज नोन एज stage. After there will be one more division. Second division, there will be formation of again 1. So here there will be 2 and 1. So again there will be 2. This condition is known as. There will be third division, resulting in the formation of 8 nuclei. So 1, 2, 3, 4, 1, 2, 3 and 4. So this is what we can write 8 stage, and then after there will be what? Next division, that is what? Cytokinesis. And if there will be cytokinesis, there will be wall formation. So here there will be movement of the cells as well as there will be wall formation. So 3 cells will remain here, the cell number 1, cell number 2 and cell number 3. So there is what? Cytokinesis. So wall formation taken place here. So we have make that again. Here there are 3 cells. So 1 cytokinesis, again cytokinesis, again cytokinesis. So 3 cells, 2. These 2 remains in the center. These 2 remains in the center. ओके, सो बेटा जो साइटोकाइनेसिस हुई है, यहाँ से आप देख सकते हैं हमने दिखाया था कि कितनी बार न्यूक्लियर डिवीजन हुआ? 3 बार. लेकिन जब साइटोकाइनेसिस हो रही है तो साइटोकाइनेसिस बेटा यहाँ से देखिए 3 सेल्स जो मूव कर रही हैं अबव पोर्शन में, 3 जो मूव कर गई नीचे की तरफ, मतलब 3 सेल्स दोज मूव टुवर्ड्स द चैलेजा, 3 दोज मूव टुवर्ड्स द माइक्रोपाइल. दे अंडरगॉन द प्रोसेस ऑफ साइटोकाइनेसिस एंड दीज 2 रिमेन्स इन द सेंटर. सो दिस कंडीशन इज नोन एज व्हाट? दिस इज रिप्रेजेंटिंग व्हाट? एम्ब्रियो sac. Let's label these 2 are the polar nuclei, because all these just made up of functional megaspore that was haploid. So ploidy level is same, haploid. These are what? Antipodals. These are also what? Haploid. This one is what? egg cell, that one is also haploid. These two are what? Synergids. Those are also haploid. So this structure is simply representing the female gametophytes. Let's count the number of the cell and again number of the nuclei. So first let's count the number of the nuclei. 1 2 3 4 5 6 7 and 8. So यहाँ पे कितने हैं? 8 nucleate. Number of nuclei 8. Number of cell 1 2 3 4 5 6 and this large cell. So how many cell? 7 celled. Okay, so the female gametophyte in the case of angiosperm is 8 nucleate and 7 celled, and such type of embryo sac is known as polygonum type. Which type? Polygonum type, because this was discovered in the polygonum plant. किसमें discover किया गया था? Polygonum plant. And remember, functional megaspores giving rise to all these structures. That's why this is also known as monosporic. What is monosporic condition? Monosporic condition means that a particular functional megaspore forming all these structure. So the process of formation of megaspore from the megaspore mother cell is known as what? Megasporogenesis. यानी एक ऐसा प्रोसेस जिसमें मेगास्पोर मदर सेल से मेगास्पोर बनता है, उसको हम कहते हैं मेगास्पोरोजेनेसिस, और यही फंक्शनल मेगास्पोर आगे चल के क्या बना लेता है? फीमेल गैमेटोफाइट. सो बेटा अभी तक हमने देख लिया ये एग है जिससे हमको मतलब तो ये फीमेल गैमेट, और मेल गैमेट तो हमने बना ही लिया था कितने? 2, जो कि कहाँ है? पॉलन ग्रेन्स के पास हैं. सो नाउ वी हैव मेक द मेल एंड फीमेल गैमेट. सो नेक्स्ट स्टेप विल बी व्हाट? ट्रांसफर ऑफ द मेल गैमेट to the female gamete, and that process is known as pollination. So I am going to erase all these things and we are going to discuss about the pollination. So this was all about the development of the female gametophyte as well as female reproductive part of the plant as well as megasporangium, that is also known as ovule. So हम अगला टॉपिक आपको बताएगा pollination. Okay बच्चों, तो अगला हमारा टॉपिक है that is what? Pollination, because we have just make the male and female gametes. So pollination is what? This is transfer of pollen grains from anther to stigma. So what is the definition of the pollination process? This is a process by means of which pollen grains, those are the male, those carry the male gametes, transfer to the female reproductive part, that is what? Stigma. Because earlier we have discussed stigma is what? Pollen grain landing site. Okay, so there are two categories of the pollination. The first one is known as what? Self pollination. आप पढ़ते आ रहे हैं इसको, that is also known as what? Autogamy. Okay, the next one is what? Cross pollination, and this one is also known as genogamy, and sometimes we can also term it what? Allogamy. Here it is clear from its name. Self pollination means that there is no need of any external agency. Cross pollination means that there is a need of agency to transfer the pollen grains from one plant to another plant, but here there is no need of what? Agency. This may be two type. सेल्फ पॉलिनेशन बेटा दो तरह का हो सकता है. एक जैसा मैंने यहाँ पे लिखा था क्या? ऑटोगैमी, एंड द सेकेंड वन इज व्हाट? जिटोनोगैमी. इसको बेटा हम लोग गिटोनोगैमी भी कहते हैं. ओके, सो लेट्स डिस्कस ऑटोगैमी. दिस इज व्हाट? ट्रांसफर ऑफ pollen grains from anther to stigma of same flower of one plant. यानी कोई एक प्लांट था, वो क्या था? बाइसेक्सुअल था. इट मीन्स दैट दिस इज पॉसिबल इन द केस ऑफ bisexual flowers only. What do you mean by the bisexual flowers? The flower that contain both male and female reproductive parts in the same. So here autogamy means that transfer of the pollen grains from anther to stigma of the same flower of same plant, and this is possible only in the case of bisexual flowers. What is jitonogamy beta? This is what? Transfer of pollen grains from anther to stigma of another flower of same plant. ओके, सो यहाँ पे क्या था बेटा? यहाँ पे अगर हम देखें सो हियर फ्लावर क्या था? सेम था. ठीक है, और प्लांट क्या था? ये भी सेम था. ठीक है, एंड हियर फ्लावर इज डिफरेंट. फ्लावर इज व्हाट? different, but plant is what? Same. Okay, so we can term it morphologically as a kind of cross pollination, but genetically this is what? Self pollination. क्योंकि अगर हम जेनेटिक रूप से देखें तो प्लांट सेम है, लेकिन मॉर्फोलॉजिकली अगर देखें तो फ्लावर चेंज हो रहा है. तो मॉर्फोलॉजिकली दिस इज नोन एज व्हाट? वी कैन से दैट क्रॉस पॉलिनेशन, एंड जेनेटिकली वी विल टर्म इट व्हाट? सेल्फ पॉलिनेशन. अगेन एग्जांपल्स हम देखेंगे, इसमें यूनिसेक्सुअल फ्लावर भी हो सकता है बेटा. In the case of unisexual flower, as well as it may be the part of the bisexual flower too, but most common we can say that unisex. This is what transfer of pollen grains from anther of one flower of one plant to the stigma of flower of another plant, to the stigma of flower of another plant of same genetic, or we can write same variety, that will be more convenient. It means that here flower is different, here flower is different, as well as plant is also different, but ये प्लांट जो डिफरेंट है बेटा, इनका जो वैरायटी है वो सेम है। ये ऐसा नहीं होगा कि कोई आंवले का पेड़ है बेटा तो वो जाके किसी मैंगो वाले को पॉलिनेट कर देगा। प्लांट का टाइप सेम रहेगा बट प्लांट डिफरेंट रहेंगे। सो दिस वाज ऑल अबाउट द पॉलिनेशन। सो वी हैव डिस्कस अबाउट द सेल्फ एंड क्रॉस पॉलिनेशन। हियर रिमेंबर, सेल्फ पॉलिनेशन डू नॉट नीड एनी एजेंसी, सो नो नीड ऑफ एनी एजेंसी, ओके। एंड हियर देयर इज अ नीड ऑफ agency. So now we are going to discuss about the agencies for the cross pollination. So we will provide all those agencies in the tubular form, then after we will discuss one by one. So I am going to make the table and then after we will elaborate. In your syllabus there are two agencies, biotic agents as well as abiotic agents. In biotic and abiotic agents, first of all we will discuss about the abiotic agents. In abiotic agents there are only two, those are wind and those are water. If the plant is pollinated by the wind, that condition is known as anemophily, and by water, that condition is known as hydrophily. And there are certain biotic agents. In those biotic agents, the first, that is most important, that is insect, that condition is known as entomophily. It may be by the animals, that may be zoophily. It may be by means of the snail, that may be termed as malacophily. So all these are the certain categories, but most important for you, that is what? Entomophily. So we will discuss anemophily, hydrophily and entomophily. Those are the part of your syllabus. So by means of a tabular form I am going to provide all those details of the agencies of the pollination, then we will discuss one by one. Okay dear students, so all these are the agencies of the pollination. First, abiotic agencies, as we discussed earlier, that is wind. This is, as you know, non-living. And anemophily, example of that is maize, palm, as well as wheat, rice, a lot of things are there. Water, if water is a medium, all pollinating agency, that condition is known as hydrophily. Example of that is Vallisneria. In Vallisneria, pollination takes place on the surface of the water, and in Zostera, the pollination takes place inside the water. Here there are certain biotic agents. In that, insects, that condition is known as entomophily. A lot of examples are there: Salvia, Aristolochia, as well as we can take the yucca flower, as well as we can also take the fig. Okay, that is known as Ficus carica. So here Salvia, Aristolochia, yucca flower, as well as there will be Ficus carica or fig. Okay, birds, that is condition is known as ornithophily, bombax and bignonia. You all aware about a particular bird that is known as hummingbird, that can fly both in the backward and forward direction, and remember it has a long beak, and by means of that it just search for the nectar and then pollen grains stick on the beak and then it carried away. Okay, next one is what? Bat. Bat is also pollinating agent, that condition is known as what? Characterophily. Kigela and Adansonia, these two are the examples of that. This one is Kigela pinnata. Okay, snail, that condition is known as malcophily, and this is simply in the Aracaceae family, or we are going to see in the case of Arisima. So these snail, bats, birds, all these are not part of your syllabus. Here in your NCERT only insect, only wind and water pollinating agencies, all these are given. Okay, so we will discuss one by one, starting with abiotic agency, and the first agency, that will be termed as what? That will be termed as what? Anemophily. And we are going to observe that what are the certain modification in the case of Animophilus flowers. Okay, so here we will discuss that what are the features of the Animophilus flowers. So feature number one, pollen grains are formed in huge amount. Okay, pollen grains beta huge amount में form होंगे, and the value of nearly that is 5 to 6 lakhs. Why pollen grains are formed in large or huge amount? Because most of the pollen grains are destroyed or waste is due to the pollinating agency, that is what? Wind. Okay, here flower is what? Open type. Open type flower means that, जिसको आपके NCERT ने बताया बेटा, chasmogamous flower. Chasmogamous flower. Okay, it means that stamen will be well exposed so that the pollen grains can be easily carried away. Here pollen grains are lightweight as well as non-sticky. Okay, as well as non-wettable, वो बेटा गीले नहीं होंगे और नॉन स्टिकी के होने के साथ साथ क्या है? बहुत ही लाइट वेट होते हैं। स्टिग्मा इज व्हाट? फेदरी। स्टिग्मा इज व्हाट? feathery. Why stigma is feathery? So pollen grains can easily stick to the what? Stigma. Here no need of nectar and not we can say that attractive color, not colorful, we can write not colorful. So all these are the advanced feature of the wind pollinating plants. In that we can take the example of maize like always, and we can also take the example of palm, and there are many more, so we can write here etc. Remember in your NCERT there is a question that is framed that why there is no need of the nectar and colorful things in those plants, those are pollinated by means of the wind and water. तो ये सारी चीजें ऐसा क्यों होता बेटा? Because there is no need of the attraction of the insect, because here the pollinating agency is what? Wind. Okay, next, hydrophily. This is very very important. This is rare. ये बेटा rare होती है और ये क्या है? Common है, ठीक है। हाइड्रोफिली जो होती है बेटा रेयर होती है। सो व्हाट आर द सर्टेन फीचर्स हियर? रिमेंबर फर्स्ट ऑफ ऑल वी शुड नॉट कंफ्यूज इन वाटर हाइसिंथ। बच्चे देखते हैं, जैसे देखा उन्होंने वाटर हाइसिंथ और वाटर लिली, तुरंत उन्होंने कहा कि यहाँ पे पॉलिनेशन किससे होगा? वाटर से। इन वाटर हाइसिंथ एंड वाटर लिली नो हाइड्रोफिली, मतलब यहाँ पे क्या होता है बेटा? जो फ्लावर्स हैं वो तो उसमें या तो शो करेंगे एनिमोफिली शो करेंगे और वो शो करेंगे बेटा इंटोमोफिली, ठीक है, लेकिन वो हाइड्रोफिली शो नहीं करेंगे बिकॉज दे आर अबव द सरफेस ऑफ द वाटर एंड दे आर वेल एक्सपोज्ड। सो वाटर लिली और वाटर हाइसिंथ में हाइड्रोफिली नहीं होता है, इसको याद रखिएगा, ठीक है बेटा। हियर ऑन द सरफेस ऑफ वाटर pollination takes place in this line is very very important, Vallisneria में बेटा क्या होता है? सरफेस पे होता है। इन वाटर होता है जोस्टेरा में, या हम कह सकते हैं अंडर द वाटर, ऑफ विच pollen grains are ribbon like। pollen grains कैसे होते हैं? रिबन के जैसे होते हैं, तो आसानी से वाटर करंट के साथ अंदर पहुंच जाते हैं। ये क्या होते हैं? pollen grains are covered with mucilage। pollen grains are covered with what? mucilage sheath, ठीक है, जिस वजह से पानी में वो लंबे समय तक क्या कर सकते हैं? रह सकते हैं बेटा, ठीक है। अब आपसे एग्जांपल अगर हाइड्रोफिली का पूछा जाएगा तो आप क्या क्या बताओगे? वालिसनेरिया, जोस्टेरा, इसके साथ साथ सी ग्रासेस, ठीक है। सो देयर आर 3 एग्जांपल्स, दोज आर गिवन इन एनसीईआरटी, बट ध्यान रखिएगा वाटर हाइसिंथ और वाटर लिली में ऐसा नहीं होता है। सो दिस वाज ऑल अबाउट 2, द फर्स्ट that was anemophily and the second that is what? Hydrophily. I will erase all these and then we will discuss about the entomophily. Pollination by insects. Okay, so क्या क्या features होते हैं? Here the flower is or petal is brightly colored. Petal is what? Brightly colored, and they have we can write scent or smell, or we can also write fragrance to attract insect. Okay, next they have nectar gland, उनके पास बेटा nectar gland होती है, that release nectar, ठीक है। nectar एक तरह का क्या है बेटा? फ्लावर जूस है जो कि क्या होता है? मीठा होता है, ठीक है। सो insects come for this. Okay, next flower is large. फ्लावर क्या होता है? लार्ज होता है। एंड इफ स्मॉल ग्रुप्ड टू फॉर्म, अगर छोटा होता है बेटा तो ग्रुप में हो जाता है और बना लेता है इनफ्लोरेसेंस। ग्रुप टू फॉर्म इनफ्लोरेसेंस, जैसा कि आप देखते हैं कहाँ? सन फ्लावर में बेटा, ठीक है। नेक्स्ट एक प्लांट है बेटा ऑफरिस रिसेंबल्स टू फीमेल मॉथ, ठीक है। अब मॉथ आता है, उसको लगता है कि ये क्या है? फीमेल है, मेल मॉथ आएगा फीमेल के चक्कर में, लेकिन एक्चुअल में वो क्या होता है? वो एक्चुअल में प्लांट होता है बेटा, ठीक है। 5वां हम देखेंगे, इंसेक्ट ले एग इनसाइड, वी कैन टेक द एग्जांपल ऑफ फिग, जिसको आप गूलर भी कहते हो, फाइकस कैरिका एंड यूका फ्लावर, एंड हेंस दे सो म्यूचुअलिज्म क्या शो करते हैं? म्यूचुअलिज्म शो करते हैं बेटा। इट मीन्स दैट हियर बोथ आर इंटरडिपेंडेंट अपॉन ईच अदर। मीन्स दैट फ्लावर में पॉलिनेशन तब तक नहीं होगा जब तक मॉथ नहीं आएगा, और मॉथ कब जाएगा? जब उसको एग को ले करने के लिए सेफ प्लेस मिल जाए। ठीक है बेटा, यहाँ से आप देख सकते हैं। नेक्स्ट, फ्लावर ऑफ एमॉर्फोफिलस कितना होता है? नियरली 6 फीट का होता है। एक्ट एज सेफ हाउस। ये सेफ हाउस मतलब 6 फीट का फ्लावर है, तो फ्लावर की साइज़ बहुत बड़ी है, तो ये सेफ हाउस की तरह काम करता है बेटा। इसी तरह से हम देख सकते हैं और भी कुछ एडवांस फीचर। दे ऑल्सो हैव फाउल स्मेल। उनके पास फाउल स्मेल भी होती है, जैसा कि हम देख सकते हैं इन द केस ऑफ रैफ्लेसिया। रैफ्लेसिया आपका टालिस फ्लावर है, तो यहाँ से आप देख सकते हैं कि रैफ्लेसिया के पास क्या है बेटा? रैफ्लेसिया के पास क्या होता है? फाउल स्मेल भी होता है। तो ऑल दीज आर द फीचर्स ऑफ द इंसेक्ट पॉलिनेटिंग plants. So now the topic pollination has 2 more things. The first, what are the adaptation for the self pollination, what are the adaptation for the cross pollination, what is the advantage of the self pollination and cross pollination. So we will quickly discuss all those. Okay बेटा, तो यहाँ से हम देखेंगे first adaptation for self pollination. Okay, तो पहला होता है बेटा homogamy. The first one is what? Homogamy. What is homogamy? Maturation of male and female reproductive part. Maturation of male and female reproductive part at the same time. एक साथ जो मेल और फीमेल रिप्रोडक्टिव पार्ट मैच्योर होती है, इस वजह से यहाँ पे सेल्फ पॉलिनेशन होता है। सेकेंड, क्लिस्टोगैमी। व्हाट इज हियर? दिस रिसेंबलिंग क्लोज्ड फ्लावर। क्या दिखा रही होता है बेटा? क्लोज्ड फ्लावर। इट मीन्स दैट फ्लावर नेवर ओपन्स। तो अगर फ्लावर ओपन नहीं होगा तो देयर इज नो चांस ऑफ द अदर पॉलन ग्रेन्स टू एंटर। अगर हम देखें बेटा, फ्लावर्स जो होते हैं इनकी 2 कंडीशन हो सकती है। एक, ये हो सकते हैं चैस्मोगैमस, मतलब ये क्या होते हैं? ओपन होते हैं, तो इस वजह से इसमें क्या होगा? क्रॉस पॉलिनेशन। ठीक है। दूसरा होता है क्लिस्टोगैमस, ये क्या होते हैं बेटा? क्लोज्ड, तो इस वजह से इसमें क्या होता है? सेल्फ पॉलिनेशन। रिमेंबर हियर दिस लाइन इज इंपॉर्टेंट। ये बच्चे कंफ्यूज होते हैं कि चैस्मोगैमस कौन है, क्लिस्टोगैमस कौन है, तो याद रखना है कि Cl और उसके बाद क्लोज। तो क्लोज अगर आप लिखोगे तो Cl आएगा। ठीक है, तो ध्यान रखिएगा, अगर Cl आ गया तो इसका मतलब फ्लावर क्या है? क्लोज। फॉर एग्जाम्पल एनसीईआरटी ने दिया है बेटा वायोला, ठीक है, ऑक्सालिस एंड कॉमेलिना। ठीक है, कॉमेलिना। सो ऑल दीज आर द एग्जाम्पल्स। नेक्स्ट, व्हाट आर द एडवांटेज ऑफ सेल्फ पॉलिनेशन? एडवांटेज में देखेंगे बेटा, no need of any agency or agent। ठीक है, no wastage of pollen grain, pollen grain का वेस्ट नहीं होता है। डिसएडवांटेज क्या है? डिसएडवांटेज ऑफ सेल्फ पॉलिनेशन, बिकॉज हियर द pollen grains is of same plant, that's why it lead to inbreeding depression। ये इनब्रीडिंग डिप्रेशन कराता है, क्योंकि एक ही तरह के पॉलन ग्रेन्स लगातार जाके प्लांट को क्या करते रहते हैं? पॉलिनेट करते रहते हैं। प्लांट्स आर लेस एडाप्टेबल। ठीक है, इफ अ पर्टिकुलर प्लांट हैव अ पर्टिकुलर डिजीज, देन डिजीज फ्री प्लांट कैन नॉट बी ऑब्टेन। डिजीज फ्री प्लांट कैन नॉट बी ऑब्टेन। क्यों? क्योंकि अगर प्लांट में बेटा कोई डिजीज है तो वो कहाँ चली जाएगी? वो चली जाएगी उसकी ऑफ स्प्रिंग में। अब हम यहाँ से देखेंगे नेक्स्ट, एडाप्टेशन ऑफ क्रॉस पॉलिनेशन। ठीक है, तो यहाँ से हम पहला देखेंगे फर्स्ट, चैस्मोगैमस फ्लावर। फ्लावर कैसे होते हैं? ओपन होते हैं, इस वजह से आराम से कोई भी पॉलन ग्रेन आके वहाँ पे लैंड कर सकता है। इसके साथ साथ दूसरा डाइकोगैमी। यहाँ पे हम देख रहे थे बेटा होमोगैमी, हियर डाइकोगैमी मीन्स दैट मैचुरेशन ऑफ मेल एंड फीमेल रिप्रोडक्टिव पार्ट एट डिफरेंट इंटरवल्स। तो सीधी सी बात, अगर एक साथ मेल और फीमेल रिप्रोडक्टिव पार्ट नहीं बनेंगे तो पॉलिनेशन कैसे होगा भाई? सेल्फ वाला क्रॉस ही होना है। ये 2 तरह का हो सकता है बेटा। एक को हम बोलते हैं प्रोटेंड्री मीन्स दैट हियर द मेल रिप्रोडक्टिव ऑर्गन मैच्योर्स फर्स्ट। बच्चे इसमें भी कंफ्यूज रहते हैं, अभी बताता हूँ आपको। दूसरा होता है बेटा प्रोटोगाइनी हियर फीमेल रिप्रोडक्टिव पार्ट मैच्योर फर्स्ट। अब ध्यान दीजिए, हमने बताया था गाइनोसियम। तो प्रोटोगाइनी मीन्स दैट अगर गायनी आ जाए तो मीन्स दैट गाइनोसियम, तो यहाँ पे फीमेल पहले मैच्योर हो जाएगा, और प्रोटेंडी का मतलब यहाँ पे पहले किसका मैच्योरेशन हो जाएगा, मेल रिप्रोडक्टिव पार्ट का हो जाएगा। ठीक है, तो इसको याद रखिएगा। हियर दिस कॉटन और ओकरा, कॉटन के साथ साथ ओकरा हम ले सकते हैं, साल्विया, और यहाँ पे हम एग्जांपल ले सकते हैं बेटा मिराबिलिस, जाल्पा, एंड वी कैन ऑल्सो टेक द एग्जांपल ऑफ रायननकुलस। तो यू हैव टू रिमेंबर, कैसे याद रखोगे, प्रोटोगाइनी का मतलब पहले गायनी है तो गाइनोसियम मतलब फीमेल रिप्रोडक्टिव पार्ट मैच्योर करेगा, और एक में फीमेल कर रहा है तो अगले वाले में मेल करेगा बेटा। तीसरा होता है यहाँ से यूनिसेक्सुअलिटी, यहाँ से इसको थोड़ा सा नीचे लिखते हैं, यूनिसेक्सुअलिटी। व्हाट इज यूनिसेक्सुअलिटी? फ्लावर हैज आइदर मेल और फीमेल रिप्रोडक्टिव पार्ट, मतलब फ्लावर के पास या तो मेल रिप्रोडक्टिव पार्ट है या तो फीमेल है। फॉर एग्जांपल इफ स्टामेन मेल रिप्रोडक्टिव पार्ट है तो ऐसे फ्लावर को हम कहेंगे बेटा स्टेमिनेट, और अगर वो पिस्टिल है केवल तो उसको हम क्या कहेंगे, पिस्टिलेट। तो ये 3 एडाप्टेशन हुए। चूंकि स्पेस कम है बेटा तो स्पेस क्रिएट करता हूँ, फिर आपको यहाँ से बताता हूँ। नेक्स्ट ओके बेटा, तो हमने डिस्कस कर लिया 3, चौथा देखते हैं हेटेरो स्टाइल मीन्स दैट हियर the size of the style varies। कहने का मतलब कोई अगर ये फ्लावर था, इसमें ये क्या थी बेटा, स्टाइल थी और स्टिग्मा, और ये आपका कौन सा था, मेल रिप्रोडक्टिव पार्ट या फीमेल। अब यहाँ से अगर पॉलन ग्रेन लैंड करना चाहेगा तो स्टिग्मा पे नहीं कर पाएगा क्योंकि स्टाइल की लेंथ यहाँ पे ज़्यादा है। अच्छा, नेक्स्ट है बेटा यहाँ पे हम देखेंगे सेल्फ इनकम्पैटिबिलिटी, मतलब pollen grains of same plant, or we can write flower will not germinate। अगर अपना ही pollen grain है तो वो germinate नहीं करेगा। नेक्स्ट एक और देख लेते हैं, हर्कोगैमी का मतलब क्या हुआ, pollen grains are enclosed in physical barrier। pollen grain एक फिजिकल बैरियर में क्या होते हैं बेटा, घिरे रहते हैं, और जब यहाँ जाके स्टिग्मा पे लैंड करते हैं तो फिजिकल बैरियर टूट जाता है। फिजिकल बैरियर कॉल्ड पॉलिनियम, एक फिजिकल बैरियर है जिसका नाम है पॉलिनियम, उसमें ये क्या होते हैं, घिरे रहते हैं। सो दिस वाज ऑल अबाउट द एडाप्टेशन फॉर क्रॉस पॉलिनेशन। व्हाट आर द एडवांटेज? बिकॉज एज यू नो हियर the 2 different plant pollen grains are combining, that's why there is a variation, and plant are more and more adaptable and healthy. So here the plants are healthy, they are more adaptable due to variation. Why there is a variation? Because here pollen grains are just landed from the different resources. What are the disadvantages? There is always need of agency, बिना एजेंसी के pollination हो ही नहीं सकता, तो ये इसका disadvantage है, और चूंकि यहाँ पे variation हो रहा है ये इसका एडवांटेज है। सो दिस वाज ऑल अबाउट द पॉलिनेशन। अब बेटा अब हमने क्या कर लिया, मेल गैमेट बना लिया, फीमेल गैमेट बना लिया। नाउ वी हैव आल्सो ट्रांसफर द मेल गैमेट टू द फीमेल वी कैन से दैट स्ट्रक्चर और स्टिग्मा, एंड नेक्स्ट दैट इज व्हाट, फर्टिलाइजेशन। तो अगला टॉपिक फर्टिलाइजेशन है। तो फर्टिलाइजेशन में सबसे पहले होता है पॉलन पिस्टिल इंटरेक्शन। ठीक है, तो अगला टॉपिक हमारा क्या है, पॉलन पिस्टिल इंटरेक्शन। पॉलन पिस्टिल इंटरेक्शन, व्हाट इज पॉलन पिस्टिल इंटरेक्शन? हियर दिस मैकेनिज्म इज जस्ट फॉर finding the correct type of pollen grains. So pollen pistil interaction is a kind of mechanism in which pistil recognize that the pollen grain that is landed over them is correct or this is inappropriate. If it is correct type of the pollen grains, means that the variety of the plant is same and quality of the pollen grains is same, then it will secrete certain kind of secretions in which there will be water and sugar that will be absorbed by the pollen grains, and pollen grains will form the pollen tube that will emerge out from the germ pore. So pollen pistil interaction actually finding the correct type of the pollen grains. If the pollen grains is not compatible, then they will distract it. If they are compatible, then they will further go the process of germination. So here what will happen? So basically we are discussing with fertilization. So here as we know in fertilization there will be fusion of male and female gamete. Okay, so how male and female gametes will fuse with each other we will discuss in certain steps. The process of fertilization was explained by the scientist Strasburger. So here we will find the concept in pollen pistil interaction. If the pollen grain is correct type, then here stigma will release, stigma will secrete, we can, that will be more convenient, stigma will secrete sugar and water that will be absorbed by the पॉलन ग्रेन, यानी जो भी सिक्रेशन किया गया किसके द्वारा, स्टिग्मा के द्वारा, उसको एब्जॉर्ब कर लिया किसने, पॉलन ग्रेन्स ने. नाउ pollen tube will emerge out, pollen tube will emerge out through germ pore, यानी अब क्या होगा, pollen tube germ pore से बाहर आ जाएगी. नेक्स्ट there will be what, growth of pollen tube, growth of pollen tube that will enter the style and will ultimately reach to the egg. It has three possibilities how. If enters through chalaza, अगर ये chalaza से बाहर जाती है, it enters through chalaza, that process is known as what, chalazogamy. ठीक है. If enters through integument, known as mesogamy, and if enters through here we can say that micropyle, that is most common, known as what, porogamy. Okay, so now pollen tube reaches in the next step, pollen tube, now pollen tube generative cell divide and forms two male gamete. Okay, now pollen tube with two male gamete, enters into embryo sac, guided by filiform. This one is important, guided by filiform apparatus. Question may be framed from here. What is filiform apparatus? Filiform apparatus is the part of synergy. Guided we can write here by filiform apparatus of synergy. Okay, now next, pollen tube ruptures and release two male गैमीट. पॉलन ट्यूब रप्चर हुई और क्या निकल गया, दो के दोनों मेल गैमीट निकल गए. हियर फर्स्ट मेल गैमीट fuses with egg, form zygote, and zygote will form embryo. Okay, second male gamete fuses with polar nuclei. How many? Two polar nuclei forms primary endosperm nucleus, that is pen will form primary endospermic cell and it will form endosperm. So this is whole mechanism of fertilization. Now let's quickly recap all those pollen pistil interaction to find the compatible type of the pollen grains. When pollen grains will land on the stigma, they will absorb the secretion of the stigma, they will grow and they will form the pollen tube that will emerge out from the germ pores, and now that pollen tube will enter to the embryo sac, and between those pollen tubes there is a callus plug that prevents the backflow of the male gametes, and during the same time here the generative cell divides mitotically to form the two male gametes, and ultimately pollen tube enters to embryo sac guided by the filiform apparatus of the synergids. Okay, now pollen tube will rupture, release two male gametes. First will fuse with egg cell forming the zygote will form embryo. Second male gamete will fuse with two polar nuclei and it will form PEN, primary endospermic nucleus, that will form PEC, primary endospermic cell, and ultimately it will form endosperm, that is what, nucleated tissue. So all these events we will discuss here by means of a suitable diagram. So I am going to erase all these things. I hope that एक जनरल सा आइडिया आपके ब्रेन में आया होगा. अब उसी आइडिया का हम इस्तेमाल करते हुए क्या करते हैं, एक डायग्राम के माध्यम से उसको बताते हैं, किसको, पॉलन पिस्टिल इंटरेक्शन के साथ साथ फर्टिलाइजेशन, जो कि आपके लिए बहुत इंपॉर्टेंट है. ओके बच्चों, तो यहाँ से देखिए कैसे होता है फर्टिलाइजेशन. दिस इज एज यू नो, दिस इज our stigma style. We will make ovary, very long portion here. This is what, ovary as you know, it attached to the thalamus, so we have making this, and inside the ovary as you know, this is what, placenta, and here there is a presence of a common, that is what, anatropous ovule. Okay, so I am just making that. This is what, typical anatropous ovule. Inside that there was embryo sac, and surrounding these embryo sac that was nucellar cell mass. क्या था इसके अंदर, nucellar cell mass. यहाँ पे क्या थे बेटा, तीन एंटीपोडल सेल्स थी, यहाँ पे 2 पोलर न्यूक्लियाई था, यहाँ पे एक एग था, ये दोनों क्या थे सिनर्जिड, और इनके बीच में ये क्या थे फिलीफॉर्म apparatus। ठीक है, तो फटाफट हम देखते हैं क्या होगा। यहाँ पे पॉलन ग्रेन आया, पॉलन ग्रेन आने के बाद क्या किया, लैंड किया। सो हियर पॉलन ग्रेन्स आर लैंडेड एंड आफ्टर लैंडिंग दे विल absorb the sugar and water. So water plus sugar, then after there will be formation of the pollen tube that will emerge out through these germ pores, and this pollen tube will ultimately entering the here embryo sac। ठीक है, तो ये अंदर की तरफ एंटर कर रही है बेटा। मैंने बताया था देयर आर 3 ways of entering this। ओके, so it has 3 ways to enter। First from the chalaza, that will known as chalazogamy, from this side that will be known as mesogamy, and from this side that will be known as what, porogamy। Now it will reaches here। Okay, so here it contains 2 male gametes by the division of the generative cells। Okay, and most importantly in between these 3 or in between these 2 lines there will be what, callose thickening। क्या होती है बेटा, callose plug or thickening। that callose plug ensure that these 2 male gametes, these are what, 2 male gametes remain together। अब हम ये वाला पोर्शन बेटा एम्ब्रियो सैक बाहर लेके आ रहे हैं। सो दिस इज व्हाट एम्ब्रियो सैक, इट हैज़ व्हाट, these 3 antipodals and these 2 are the polar nuclei, and here this one is egg and these 2 are synergid, और इन दोनों को हम क्या बोलते थे बेटा, combinedly known as egg apparatus। तो ये क्वेश्चन पूछा जा सकता है, what is egg apparatus? So synergid along with egg is known as egg apparatus। Here there is a presence of filiform apparatus। filiform apparatus जो है आपका, this will guide the pollen tube। Okay, now we will just make this portion only। So this is the wall of the embryo sac। Okay, these are the egg cells, these 2 are the synergids, and here there is what, filiform apparatus। Now pollen tube just coming, it will reach here, and as you know pollen tube has callose plugs, so we will make those callose plugs। These are the 2 male gametes। Now this synergid will start degeneration, and after starting degeneration this filiform apparatus will guide this pollen tube to enter the embryo sac। Now this pollen tube will enter the embryo sac and will rupture, and it will release these 2 male gametes। What will happen here, as you know there are 2 polar nuclei, so here this one is the first male gamete, it will fuse with this egg cell। Okay, and this will form what, zygote, and zygote will later on form embryo, और फिर आपको मालूम है कि सीड वगैरह का फॉर्मेशन होगा। दिस वन इज द सेकंड मेल गैमीट। सेकंड मेल गैमीट विल फ्यूज दिस 2 polar nuclei। so this one is N, and N means that this one is haploid, this one is also haploid। so the first male gamete that was haploid fuses with the egg cell that was also haploid, so forming the here diploid zygote। Okay, second male gamete that is also haploid, so N, these 2 N plus N plus N, it will form PEN, primary endosperm nucleus, it will change into PEC, and that will form endosperm। so this is what whole process of fertilization। now here fertilization takes place twice। first male gamete fuses with the egg cell, fusion number 1, second male gamete fuses with the 2 polar nuclei, fusion number 2। so that's why this is also known as double fertilization। so what is double fertilization? this one is when the first male gamete fuses with the egg cell, that was known as syngamy, and here because N plus N plus N it is thrice, that's why triple fusion। so here we can write syngamy that forms zygote plus triple fusion that forms endosperm is equal to what, double fertilization। so question may be framed as what is double fertilization? so syngamy plus triple fusion is known as double fertilization। you can make this diagram also, you can mention that first male gamete fuses with the egg forming the zygote, that will form embryo, second male gamete will fuse with 2 polar nuclei, it will form PEN, primary endospermic nucleus, that form PEC, primary endospermic cell, that will form endosperm। so this whole process is known as what, double fertilization। so this is shown here, fertilization as well as double fertilization। so now all the topics those were fertilization as well as double fertilization are completed। so fertilization is the fusion of male and female gamete। so this one is first male gamete with the egg cell is fertilization, that is also known as syngamy, second male gamete with the 2 polar nuclei, that is known as triple fusion। so syngamy plus triple fusion is equal to double fertilization, because here fertilization takes place twice, एक male gamete का egg cell से, फिर दूसरे male gamete का polar nuclei से, इस वजह से 2 बार fusion हुआ, तो double fertilization। so this was all about pre-fertilization structure and event। so in this video we will discuss only pre-fertilization structure and event। in the part number 2 video we will discuss about the post-fertilization structure and events। so because this video is already nearly more than 1.5 hours, so we will not just conclude all those portions. So we will make a separate video, and that will be part 2 video. So thank you very much for watching the video, and I hope that after watching the video there will not be any doubt. So if you are able to clear your concept, don't forget to like the video and subscribe the channel and share with your friends too. Okay, and most importantly, from this topic, questions will be asked in your board examination. So wait for the next video, that will be part 2. In these 2 videos we will discuss whole the content of the chapter number 2. Actually there was a chance that we are going to complete the whole content in a particular single video, but that is not possible. वो possible नहीं है बेटा, क्योंकि आपके concept को भी clear करना है और एक एक चीज़ें आपको clear करते हुए चलनी है. So it is not possible. That's why I will make the part number 2 video, and एक chapter 2 video में भी अगर complete हो रहा है तो that is much more important for that. In part 2 video we will also discuss about the important questions. Okay, so don't forget to watch the part 2 video. So that is all about for today. Thank you very much. Jai.